Hello and welcome back, students, to the second lecture of Class 10 Biology, Chapter Life Processes. In the previous class, we studied about characteristics of living things. We studied about types of the various living things. We also studied about the various life processes which we maintain by which we maintain our lives. We saw that basically four important life processes are responsible for maintenance of our life as per our syllabus right now. Here we saw that mode of nutrition can be autotrophic or heterotrophic. We also saw that most of the animals or the living things are actually not able to produce their own food, so they are put under the category of heterotrophic animals, and they are called heterotrophs, and the nutrition is called heterotrophic nutrition. So living things which cannot make their own food are called heterotrophic animals, and they depend directly or indirectly on green plants for food. Based on the feeding habit, living things are classified into various kinds. We know that the various types of heterotrophic nutrition can be classified under following headings. Number one is saprotrophic nutrition, where the living things absorb liquid food from the surface of dead or decayed organic matters. They actually absorb the liquid food through their suckers. They digest the food outside their body, and hence they are called saprotrophs. What does it mean they digest the food outside their body? Actually here they secrete certain enzyme on the surface on which the food is present, and there only the food is digested, and when it is ingestible or means able to be taken in, they are sucked up through the suckers. This happens in the bread molds, this happens in the mushrooms, and some other fungi which you can find out as per, because you had studied this fungi kingdom in class 9th, diversity of living organisms. So you can relate those things very fresh in your memory, so you can find it out. They are called saprotrophs. Generally, there is another group of living thing which absorb liquid or digested food from the living organisms. Those are called host, and the living things who are absorbing are called parasites. So the nutrition is called parasitic nutrition. In plants, actually there are two kinds of parasites which are totally dependent for the food on the other plants. They are called total parasite, and some are actually only dependent for raw materials to produce their own food, so they are called partial parasites. But in animals there are parasites which live inside the body, so they are called endoparasite, and the parasites which live outside the host body is called ectoparasite. Here examples of the parasite are mentioned as far as possible: cuscuta, leeches, lice, orchids, etc. are given as well. Many examples you can also find out in as per your awareness. Here the third category of heterotrophic nutrition can be holozoic nutrition. Actually, my dear children, holozoic nutrition in the unicellular is of different kind, in the multicellular is of different kind. In the unicellular organisms, it is done through cilia, through flagella, through pseudopodia. So one example is mentioned over here, amoeba in the unicellular. One example is paramecium in unicellular, which uses cilia and trichosis to kill the food and absorb the food. We had studied these things in class 9. On the other hand, actually the most important feature that makes them distinct from other category of nutrition is that they digest their food inside their body. Once they take in and they then digest. So there are 5 steps of holozoic nutrition we will take up in the next class. Please stay tuned to the channel, and don't forget to subscribe and share. Well, they digest their food inside their body. Example: amoeba, paramecium, humans, fish, etc., and many other animals you might have seen around. So they may eat plants or animals or both. They may eat according to their feeding habits, they can eat their different kinds of foods. Let us look at the different types of animals in the multicellular category who are having holozoic nutrition. Here the holozoic nutrition can be found in the three different kinds of animals like herbivores. They eat plant or plant products. We all know this very well. They have all well developed broad grinding teeth at the jaw, in the jaw, upper and lower jaw. There are incisors and premolars and molars. So they are well developed so that they can chew the grasses and leaves properly. Actually, their small intestine, which is the final place for the digestion of food, that small intestine is longest in length in the herbivores. Why? Because they have to chew cellulose. So digest cellulose. Cellulose is a very complex kind of carbohydrate and it needs a longer time in the small intestine. So longer time in the small intestine and also the longer surface area. So in order to get digested properly, they should stay for a longer time in the small intestine. So for that purpose only the herbivores have got adaptation of longer small intestine. In contrary to this, carnivores have got habit of eating flesh of other animals, which is softer and less complicated in comparison to the cellulose. So they have actually, first of all, the pointed teeth well developed, which are called canines for tearing flesh, and their small intestine is comparatively shorter. Well, omnivores, since they eat both plants and animals flesh, so they bear the facilities both of herbivores and carnivores. Apart from that, think about what about their small intestine. Well, you can see the figure here, which category does it belong to, which category does this belong to, and what would you feel here. Well, let's see the other thing. Elaboration of the nutrition in plants can be done in certain points. It is very important for you to note that if you write points in the description instead of paragraph, then it becomes very clear and distinct for you. So the bullet and numbering method can help you a lot in maintaining the notes of these topics. So here as we all know that this pictorial presentation you relate, this was shown in the previous class. So let's analyze this whole picture with the help of certain points. Number one, we know that plants can synthesize. Well, children, here you have to understand that production is different from synthesis. Well, production means something which was not there and it is produced. Well, synthesis is something which was present in certain other forms. Now it has been converted or combined. So here what was present earlier, here they will deal with this later on. So synthesis, they synthesize their own food. Synthesis means combine. So the process is called photosynthesis. Photo means light, in the presence of sunlight, and synthesis means to combine. So that's why the process is called photosynthesis. What do they need? Certain raw materials. The raw materials what they need are carbon dioxide, which is available in air, and the water from soil as raw materials. They perform it is in the presence of sunlight and chlorophyll. Where is chlorophyll? We will see later on in the detail in the later part of the slides. Well, sunlight and chlorophyll act as catalyst and support here. Well, they produce food in the form of glucose and they store the food as starch, as a reserved food. We will talk about this later on. My dear children, if we talk about the steps of this photosynthesis, it occurs in 3 steps. First of all, you check out this chemical equation, where the first group is called reactant and other group is called the product. In the first group, the reactant, we find the carbon dioxide, other find in the water. These are the 2 raw materials that plants need, and in the presence of sunlight. In the presence of sunlight, what do? They convert the carbon dioxide and reduce it to the form of glucose. What is the definition of reduction? Definition of reduction is addition of hydrogen and removal of oxygen or removal of oxygen. So you can check out the number of oxygen in the left hand side and number of oxygen in the right hand side in the main compound. Apart from that, there is a production of oxygen as a byproduct. Apart from that, water is also released. So how? Let us see. So you know that there is a green pigment of chlorophyll. This green pigment chlorophyll has got very important functions to play or role to play. That is, first, they help in absorption and trapping of the sunlight. It is done by the chlorophyll. So chlorophyll plays a very crucial role here. Apart from this, chlorophyll also does one important thing. They convert the light energy into chemical energy. And not only that, they help in the sunlight to break water molecules. What is water made up of? Water is made up of hydrogen and oxygen. So that molecule of water is broken into hydrogen and oxygen. So it is this oxygen which is produced as the byproduct, and that what we inhale in the air, inhale from the air. So this is oxygen as a byproduct. Apart from that, reduction of carbon dioxide. This carbon dioxide, which had 12 oxygen here, that gets about only 6 oxygen, which had no hydrogen, it becomes the 12 hydrogen. So that's what definition of reduction is justified in this chemical equation. You can see that. So in this way, the carbon dioxide is reduced into glucose, right? C6H12O6. That's called carbohydrate. That's a form of carbohydrate, right, children? So here, in summary, it is said that photosynthesis is a process in which the carbon dioxide and water combine in the presence of sunlight and chlorophyll to produce glucose. Apart from that, oxygen and little bit of water vapor. Well, after that, in order to study this in detail, let us see some important pictures here, my dear children. You know that which part of the plant is called kitchen of the plant? Kitchen of the plant is the chlorophyll, whereas the food factory is the leaf. What part of the leaf is important for the food production? That is the chlorophyll. Now, where is chlorophyll present? The chlorophyll is present in the cell, present in the lower surface of the leaf. And that's what we will find out in the entire leaf. You will see, first of all, you can see this. Let's see first this picture. If you cut the leaf into certain the cross section, then if you cut a leaf into the transverse section, you will get to know something. What is that? Let us see. Well, technical issue. This is the leaf picture. In this leaf picture, you can see that the leaf was cut from this area. When the leaf was cut from this area, then the lower surface and the upper surface, this is the midrib of that. There is labeled as a phloem, vascular bundle, xylem bundle, right? So in this section, if you take up the section of the leaf here, then you can check out. This is the upper surface of the leaf. This is the upper surface of the leaf, like this. So in the upper surface of the leaf, you will see the upper epidermis, right? This is the upper epidermis. And not only this, here you can also find out that this is the lower surface of the leaf. This is the lower surface of the leaf. The lower surface of the leaf is lower epidermis, and this is the upper epidermis. The upper epidermis of the leaf, which is covered with the cuticle. This is the part of the leaf which is important for you to study. Upper epidermis has the oily waxy surface. This wax cuticle is this. Then the lower epidermis is covered up to here. Then there is the cylindrical lines. This is called, as you call it, palisade mesophyll. Palisade mesophyll, these are the columnar kind of cells which provide in the surface area. On the other hand, you can see this air space. This air space is actually, this air space makes this whole cell spongy. So that's why it's called spongy mesophyll. Not only this is spongy air space is continued till outside of the lower surface of the leaf. So outside of the lower surface leaf, this creates a pore. This makes up a pore. This pore is helping in the exchange of gases. This becomes stomata. What does it become? Stomata. And not only this stomata, along with the some cells, right, here in the lower surface, that this is all present here. This all actually form a lower epidermis. If we talk about the next important thing, that this is the pore which we were talking about in the previous slide, right? So we will see one by one. So if we talk about this slide here, so in this we can see that green pigments which are present in the leaves, Green pigment which are present in the leaves are called chlorophyll. It traps the sunlight and converts solar energy into chemical energy. It helps in breaking of water into hydrogen and oxygen. They also help in reduction of carbon dioxide into glucose. We had studied this. So it shows that here, my dear children, this internal the leaf structure, this shows that the chloroplast contains chlorophyll. The chlorophyll, what is that? This is one. Well, check out this. In this, if you study the chlorophyll, then you will see it's made up of this kind of thing. First is outer membrane, outer thicker membrane, you can see. The second is the inner membrane, intermembrane space. Between this and this there is a space, and then this comes in the inner membrane. So from here to here, the entire space is called intermembrane space. Well, inside the chloroplast, you will see that this is the stroma. The stroma means what? The entire area is filled up with aqueous substance. This is a fluid is called area is filled up of. This entire area is like a cytoplasm of cell. So the fluid is filled up in this area. This area is called stroma. And apart from this, there are some coins like kept upon one another, piled upon each other. This is piled upon structure are called granum. So there are two basic major structures present inside the chlorophyll. One is the stroma, which is the liquid part, and the grana is the which is stacks of. What is this actually piled up structure? This piled up structure is called thylakoid. The piled up structure is called thylakoid. This is granum and this is the aqueous area stroma. Not only this, apart each and every piled up structure is actually connected with certain things. So that connection of membrane, that through the membrane, this is called lamella, right? And if you see this entire the circular, if you check out and see that like a pipe, if you see from here to this longitudinally vertically, if you see, then the entire the hollow surface area entire is called lumen inside the thylakoid. This picture can be paused and seen again and again in the video. All right. Yes. So chlorophyll is this is chlorophyll. If you check out the cells in the plant, then you have to recall your class 9th concept. In any part of the plant, the cells will be similar kind of structure will be found. That one is the cellulose cell wall, next the cell membrane, then vacuole which is bigger and central, then the peripheral nucleus, entire this area, open area which is filled up with the cell organelles which is called cytoplasm, and here inside the cell only you will find one plastid, right? The plastid, out of 3 plastids, one plastid which is green colored pigment is called a chloroplast. So the chloroplast is magnified in this. So that's why the next slide was shown with the chloroplast. Once more you have to relook into these slides one by one. This all the structures in one slide are here which are related with the photosynthesis. One thing, this thing, this thing and this thing. So here this one can be seen once again in this slide, my dear. What is this? This is the structure of a leaf once again, for which we can see this the upper epidermis. This is the upper epidermis, and columnar, these are the palisade parenchyma, palisade mesophils. Palisade mesophils के बाद आपको ये area आता है जिस where the air sponges and all the air spaces are present. So air spaces are present and making this whole area less solid, less dense. That's why they are called spongy mesophyll. Well, we are not concerned about that right now. We are concerned with the pores which are present in the lower surface of the leaf. These pores of the leaf are actually accompanied with these structures. First you observe this, the entire arrow mark. This is the when the higher water vapor content is there. We will talk about this in this slide one by one. What are these we are talking about? These are stomata. Stomata are tiny pores present in the lower surface of leaves. What do? They allow exchange of gases and helps in transpiration. What is transpiration? In class 7 concept we recall it, that transpiration is a process by which leaves release water vapor through the stomata. So that's what is transpiration is there. The transpiration. What is stomata made up of? Stomata is actually made up of certain parts. Let us see. Check out. Here all the pictures are given in one group. Here it is distinctly shown one by one. So if you remember the shape of two kidneys, so like that two kidney shaped parts can be seen. This is called guard cell. What is it called? Guard cell. This guard cells have got one important feature. What is that? They contract and relax. So when they expand and contract, then that is actually the main factor by which the stomata stomatal pore opens or closes. So its opening and closing is controlled by the guard cells. What makes the guard cells to open and close the stomata? There is one important factor which is called pressure. Pressure built by what? Pressure built by or due to entry and exit of water. Where from? Water comes from the roots and they come to the stomata. When it comes to the stomata, then actually these guard cells are also accompanied with the help of some epidermal cells. You again go back to the recalling concepts of the previous class. So there are two parts in the stomata basically. One is the stomatal pore, that's called stoma, and these are the two bean shaped guard cells. And not only this, inside the guard cells there are chloroplasts also present inside that. Well, an outer cell layer of the cell, which is, you remember, that's the epidermis, right? Each and every epidermal cell is accompanied with nucleus. You can see, check out this, right? So water, actually what does water do with the stomata? Water, when enters through the process of diffusion, then that creates a pressure on the guard cell. The guard cell expands. When the guard cell expands, then it creates a pressure on the stomata, then stomatal pore opens, right? And when the water is out, so when the water is no more present inside that, then it contracts. So water diffuses into the guard cells, which causes them to open. On hot or dry days, the guard cells have less water and they relax. When they relax, the stomata closes. So you can just imagine you join two palms together, and once you straighten the palms, the stomata closes. Once you make them curved, then stomata opens. This is all happening due to the turgor pressure built up by entry or exit of water into the guard cells. So swelling of guard cells causes opening of stomata, and drying of guard cells or contraction of the guard cell causes closure of stomata. So when does the stomata opens a lot, or the opening and closing of the alternate time of the closing and opening, it becomes faster? When it happens in the summer season. When the season is dry, then rate of evaporation of water is more, rate of transpiration is more, so the opening and closing of the stomata becomes faster in the plants, right? Faster in the plants. Why faster in the plants during the summer season? Because the rate of transpiration is higher, rate of evaporation of water is also higher due to that only. In the night time, the sunlight is not there, so stomata mostly closes. In the daytime, its opening and activity becomes more frequent. All right, children, this is what about the stomatal behavior. So this stomata has got, if you could see this, the parts are, these the different parts are shown here in detail, which is not in your syllabus, but for the illustration it is given here like this. So diffusion of water in this has been given show very clearly. Well, my dear children, before we go ahead, we must check out one thing, that before we go ahead, we must look into this part once. How to check out whether the leaves are producing food or not. Plants can synthesize their own food, we all know that, but how do we check whether the food has been prepared or not? So recall your page number 96 in your book. Once you can check out this activity is number 6.1. In this, if you take a potted plant with variegated leaves, in this, for example, any money plant or crotons. So if you take this plant, keep the plant in a dark room for 3 days, so that all the starch gets used up. Now keep the plant in sunlight for about 6 hours. Well, pluck the leaf from the plant, mark the green areas in it and trace them on a sheet of paper. Now here you can see that dip the leaf now in the boiling water for a few minutes. After this, immerse it in a beaker containing alcohol. Then you can see that carefully place the above beaker in a water bath and heat till the alcohol begins to boil. What happens to the color of the leaf? What is the color of the solution? You have to note down later on. This is the part of practical, actually you have to do it till the alcohol begins. Now, so color of the solution. Now dip the leaf in a dilute solution of iodine for a few minutes, right? Take out the leaf and rinse off the iodine solution. Observe the color of the leaf and compare this with the tracing of the leaf done in the beginning. What can you conclude about the presence of the starch in the various areas of the leaf? You can check out the figure number A, what is the color of leaf here, and figure number B, what is the color of the leaf here. You can also check out the variegated leaf A before and B after test, right? So it's a very important part of the practical question, and you have to remember this. And please observe this very clearly, because every year one or two questions, one question in the practical category, they are asking related to this experiment. This experiment is very important. So if you carefully observe the cross section of the leaf under the microscope, you will see this part. So after this, if you go through the activity, then you will see you will see that this part of the leaf is getting, well, there is a technical issue, please bear with us. So this part is this leaf, when it is dipped into the, if you remember the class 7's concept, in which we, after the starch test is done, what is the change in the color? So that change in the color can be visible in the particular leaf. When the leaf was not done with the starch test, this is the behavior, and when the leaf was done with the starch test, this is the behavior. You have to observe this, and please remember. Now after this, in order to check something more, we must also take a great stock of this picture of the stomatal pore once again, because NCERT is the base of your knowledge. So the whole classroom teaching is not enough until unless you go back to the book and relook into the concept. So in that you will see that the massive amounts of gases exchange that takes place in the leaves through these pores for the purpose of photosynthesis. But it is important to note that exchange of gases occurs across the surface of stems, roots and leaves as well. Since large amount of water can also be lost through the stomata, the plant closes these pores when it does not need carbon dioxide for photosynthesis. So here this closing and opening of stomata is important, right? It should not remain always open, it should not remain always closed also. So what controls? That's where the role of guard cell comes into play. So that's what causes the stomatal opening and closing. That is due to the control on the guard cell swelling when water flows into them, and shrinking when the water gets out of them. Here another activity is important in your syllabus to check out, because this is also, these two questions are very important when it is asked in the in the exams. So this activity will be seen in the next episode of this classes. Well, stay tuned for the next video soon to be uploaded in this series, my dear children. So today we come back to the recapitulation of this today's class. We come back to the recapitulation of today's class. We studied in this class the nutrition, mode of nutrition, where recapitulation was done for this heterotrophic mode of nutrition. Then we saw this nutrition in plants and plants. Here we saw that the plants can synthesize their own food. They do it in the presence of sunlight and chlorophyll with the help of carbon dioxide and water as raw materials. So since there is a combination of carbon dioxide and water in the presence of sunlight and chlorophyll, so this is called photosynthesis, and here they produce food as glucose and store as starch, right? 3 steps of photosynthesis. First of all, absorption of sunlight by chlorophyll, then conversion of light energy into chemical energy, breaking of water molecules into hydrogen and oxygen, and third step is the reduction of carbon dioxide by hydrogen into carbohydrate, right? Next we see that what are the various factors that helps the plant to produce food. The first important factor is the chlorophyll. What happens when chlorophyll is not there? You can see in the picture of variegated leaf, right? The chlorophyll is a green pigment present in the leaves. It traps the sunlight and converts solar energy, which is already shown in the previous slide. It shows the chloroplast contains chlorophyll. Stomata is present in the lower surface of leaf. This is the all-in-one diagram in the one slide. One by one, these diagrams are shown in the detail, where the leaf structure is taken as a cross section is seen. So upper epidermis, lower epidermis, mesophyll. Mesophyll is into two parts. One is the palisade, which is columnar, and gap. But where the area space is present, it becomes a spongy. So it becomes a spongy mesophyll, where the wax cuticle is present in upper layer also and in the lower layer also. Lower epidermis contains stomata. Alright. So here this is a typical plant cell, which is actually containing plastid. Plastid, out of many plastids, one is the chloroplast, which contains chlorophyll. The chlorophyll looks like this. In the chlorophyll you will see the thylakoid and grana. Thylakoid, stoma, grana. Thylakoid, stoma, and grana. So this area, open area, is called a stroma, and this area is called granum. Granums are piled up with thylakoid, and these are connected with the membrane called lamella. The hollow area is called lumen. Outer membrane, inner membrane, intermembrane space. Alright, yeah. Here leaf structure is again relooked into: upper parenchyma, upper epidermis, palisade mesophyll, spongy mesophyll. हाँ, अब क्या होता है यहाँ पे? You can once again really go into, when the higher water vapor content is present inside the leaf, then the expansion takes place, and then the vapor gets out, the guard cell opens, and and when the low water vapor outside, then what happens? It takes a higher concentration to lower concentration. You recall the concept of diffusion. So when the water vapor concentration inside the leaf is high, it will move out, and if it's higher outside, it moves in, right? Out of similarly, at the same time, when the stomata opens, then the carbon dioxide, as per the content, moves out or in. Okay, so this is the movement of carbon dioxide from the higher concentration to lower concentration, and alternate opening and closing of the stomata is actually helping in allowing of the carbon dioxide movement into the and water vapor to leave. This leaving of water vapor and entry of the carbon dioxide is actually done with the help of stomata. All right, so this is the relook into stomata. Stomatal pressure, stomata contains the guard cells and stomatal pore. The whole is called stomatal apparatus. This is all due to the turgor pressure created by entry or exit of water. That depends on the temperature, that depends upon the speed of air, that depends upon the rate of evaporation, the hence rate intensity of sunlight, are the various factors that creates the opening and closing of stomata frequency, right? Thank you. This is all for today. Stay safe and keep educating yourself. And please follow the channel so that the next video notification goes to you. Please give your review. How was the video if you like it? Thank you.